Hi, welcome to section B. We are going to look at fatigue and how the body recovers from exercise throughout section B. This is section B1 where we're looking at the causes of fatigue, and we will be looking at section B2, the recovery of energy systems, in the next video. We will look at nutritional strategies to help recovery. We're going to look at recovery of the musculoskeletal system, and finally section B4 we will look at overtraining and the issues that occur with overtraining. And that's it for section B. So let's make a start. First of all, we all know that we can't exercise forever, and I've used the classic example here with the Brownlee brothers in the World Championships. We need to look at physiologically why our bodies are forced to reduce the intensity of our exercise. So in the picture there and in the situation here, Jonathan Brownlee just slowed down. His body forced him to reduce the intensity in which he was working at. Why does that happen? Well, it happens for three reasons: a build-up of waste products, depletion of energy sources, and also depletion of acetylcholine. And the next few slides will explain those 3 points in a little more detail. So if we think of exercise as anything beyond resting levels, so anything above that of sitting in a chair relaxing perhaps, so our body is working harder to provide the energy for muscle contractions. Our body will eventually prevent that repeated muscle contraction, and it will stop even the electrical impulses being sent to the muscles to contract. When we get to this stage, in order to recover, we must rest. So in section A7, we looked at energy stores, and we talked about the store of glycogen in the liver and the muscles. We looked at stores of phosphocreatine in the muscle cells, and we looked at the use of adenosine triphosphate as our source of energy for muscle contraction. So if we think about exercise and potential situation for using a high intensity for perhaps a prolonged period of time, so a marathon run, or it could be that we have played a game of football, so we've done repeated high intensity for 90 minutes, we would be assuming that our glycogen levels have depleted or at least reduced, our phosphocreatine has reduced, and therefore the amount of ATP that we have readily available for that first couple of seconds of exercise will also be reduced. Fats and proteins are not depleted after just a normal game of football or even an ultramarathon for that matter, as it is very, very energy rich. So it would take a very long time before our fats would become depleted. Protein is not depleted either, as if you remember, we didn't use protein for energy unless we were in a life or death situation. So let's look at a bit more detail of these energy sources that we looked at in section A7. And first we'll have a look at glycogen. We know that glycogen is stored in the muscles and liver, and approximately 350 grams is stored in our muscles compared to 100 grams in our liver. It can take about 2 hours of exercise to deplete those glycogen stores. So we're talking fairly high, moderate to high intensity exercise will deplete the glycogen stores. So if we were to run for 2 hours, then the glycogen stores in our muscle and our liver will have depleted. Glucose is the smaller version of glycogen. So it's broken down from glycogen, if you remember, and that is in the bloodstream. So it's not a stored version, but it is moving and travelling in the blood. And there's only about 20 grams in the blood at any one time, which we know is delivered to the muscle cells and then used for resynthesis of ATP. Phosphocreatine is stored in our muscles and referred to in this chapter as one of the phosphagens. So if we see the word phosphagen, it's referring to either phosphocreatine or adenosine triphosphate, both being the phosphagens. So phosphocreatine is made in the liver, and we know is stored in the muscle. And we also know it lasts between 8 to 10 seconds of very high intensity activity, so slightly different to glycogen. ATP in the muscle cell that's there ready for us the minute we stand up there's about 2 seconds worth in the muscle cells before it runs out. It's important to note that fat and glycogen is usually metabolized together. So glycogen being the predominant source for the higher intensities. And the reason for that is that glycogen was quicker to break down than the fat. If you remember in the lesson, we talked about glycolysis happening and then going into the Krebs cycle before it goes into the electron transport chain. Fats are sourced from under the skin in the adipose tissue, which is not what you need to know for your specification, but just for information. And it comes into the muscle cells still, or I should have drawn that round there, apologies, where glycolysis occurs and where the Krebs cycle happens, but it misses glycolysis as that's the partial breakdown of glycogen. It requires lots of oxygen to break down the fats into fatty acids. It goes under a process called beta oxidation. Again, not in your specification, but interesting to know. It takes a huge amount of oxygen and quite a lot of time, as there's a lot of changes, more so than there are in glycolysis, before it is allowed to go into the Krebs cycle and be metabolized through the aerobic system. So you don't burn the fat alone. The glycolysis system will always be breaking down glucose from the glycogen stores all the time. If we get to a situation where we've run out of glycogen, then the whole system will slow considerably, and you will get a situation of Jonathan Brownlee or of the ladies we watched doing the Ironman competition. So we've talked about depletion of energy stores, and now we're moving on to the second cause of fatigue, which was accumulation of waste products. So in moderate to high intensity, we know that lactate and hydrogen ions can be produced, and that came, if you remember, from our A7 lessons. We have glycolysis and we get to a substance called pyruvate. And if there is not enough oxygen to join with pyruvate, so we're running out of oxygen, this then dissociated into lactic acid, which went into the bloodstream as lactate and hydrogen ions. If you're not sure and can't remember that, then it might be worth going back over A7 video. So what does this cause? It causes the blood pH to be reduced. Now, blood pH is anywhere between 6.4 and 7.4 is fine. If the pH goes lower than 6.4, then the body will start to stop muscle contraction. So this point, interruption in muscle contraction. It will prevent the muscles from contracting by affecting the neural function. This is the neural function here. The message, the electrical impulse being sent to those muscle fibres to contract, that will be interfered with and won't happen, or won't happen as efficiently as it did happen before, if the pH is lower than 6.4. During low intensity exercise, we create carbon dioxide, as you know. So here we've got pyruvate, we've got oxygen, but instead of going out into the bloodstream as lactate and hydrogen, we've got this oxygen coming in, and we can go to acetyl coenzyme A and around the Krebs cycle. And if you remember, one of the byproducts of the Krebs cycle was carbon dioxide, which goes out into the blood system. So that's where we get it from. Now, it travels in the blood as carbonic acid. But what we haven't talked about is this. Carbonic acid can travel in the blood. If there is too much carbonic acid in the blood, it will dissociate. That means it splits, and it splits into bicarbonate and hydrogen ions. And there we have our problem. We've now got more hydrogen ions. So this doesn't happen immediately, but this happens after a prolonged period of low intensity exercise, such as running around Carsington Water or doing a marathon run. So with any moderate to high intensity activity that you've done, maybe a hill or maybe you've overtaken someone on the marathon, you're going to produce lactic acid, which dissociates to lactate and hydrogen. And now with any of your low intensity or aerobic exercise, the carbon dioxide is now not able to get out or travel in the same way because we've got too much carbonic acid, too much carbon dioxide being produced because the aerobic system is working flat out. that it now dissociates and we get even more hydrogen ions. If we're getting hydrogen from both, it is likely that that pH is going to reduce quite quickly and further stop this neural signal. One final point to make on this is that if we have a decrease in pH, it also stops the release of metabolic enzymes. Well, metabolic means, in this case, it means these energy systems here. Enzymes are those chemicals that make things work, that make things change, and these are used all the way through glycolysis. They're used around the Krebs cycle, they're used to break down the glycogen. At the beginning, they're used to break down fat. So if we lose those metabolic enzymes and they're no longer released because of the reduction in pH, then we are going to limit the amount of ATP we can resynthesize. If we limit the amount we can resynthesize, we limit our muscle contraction. Therefore, we go back to the first slide, our body is forced to reduce its intensity. So the final part is depletion of acetylcholine and calcium ions. So what is acetylcholine? First of all, we need to talk about that. It's a neurotransmitter. Now, a neurotransmitter, if you remember, is stored around here at the end of the motor neuron. So it's at the junction between the motor neuron and the muscle fibre. If there are lots of neurotransmitters present, then the electrical impulse can carry across from the motor neuron and contract all the muscle fibres that neuron is attached to. Now, if you remember section A6, the endocrine system, we looked at the impact of testosterone. And if we are working on weight training and things like that, testosterone can increase the neurotransmitters at this junction, which meant we could get a more powerful contraction, or we could be more responsive to certain movements, certain powerful movements. So acetylcholine is one of these neurotransmitters that we need to transmit that signal. If acetylcholine is depleted, so we don't have as much of it, then we are not going to be effective at sending that electrical impulse across. If we can't send that electrical impulse across, then it means we cannot contract our muscle fibres. The next part of this was the calcium ions. Now, you will learn a lot more about this in Unit 2, where you will learn the structure called the sarcoplasmic reticulum. That's what this SR stands for. Now, this is where calcium is released from. We don't need to know that for Unit 1, but you do need to know that calcium is used for muscle contraction. In Unit 2, you will become aware that calcium is released by the sarcoplasmic reticulum, and it is used for the myosin cross bridge, which is where the actual movement occurs of these muscle fibres. We need to know for Unit 1 that calcium is depleted. If calcium is depleted, it can't be released into these muscle fibres, and if it can't be released into the muscle fibres, then we cannot contract. So we've got two reasons here, another two reasons here why we can't contract our muscles, or reasons as to why our body suddenly flakes out on us and forces us to reduce our intensity from a jog down to a walk. So to recap, you've got the depletion of energy sources, we've got depletion of acetylcholine and calcium, and we've got the accumulation of waste products, which all ultimately end up as hydrogen, but they start as carbonic acid with carbon dioxide or lactic acid. That's it for section B1.